Hi again. In this video, I will explain you more details about QEUCHERS method for extraction pesticides and food samples. QEUCHERS method is a very easy method that it can employ in any lab. I would like to explain to you about this method, and as you know, this method involves different steps, especially extraction steps, and also cleanup steps. I will show you what to do and how we can do this method. Okay, this is terphenyl phosphate, which is used for internal standard. In the first step, after spiking the sample, if you have need to do this step, then we make a solution of this internal standard with a concentration of 5 milligram per liter. We can add this internal to your sample. You have prepared this sample with grinding the sample. It's good to mention that when you have the food sample, in the first step, you should put the sample in the freezer for about 30 minutes, and after that, grinding the sample and weight the sample depends on the matrix that you have. For example, for cereals, we use 5 grams. For fruits and vegetables, we use 10 grams. This is a wheat sample. I have weighted 5 grams. In this step, I add 200 microliter from TPP with a 5 ppm concentration, as you can see. Okay, I will add the internal standard to the sample. Then we shake the sample and vortex it and put it for about 5 minutes. After that, we will start the next step. We can vortex the sample, as you can see, then put it for 5 minutes. Okay, after 5 minutes, the water, deionized water should be added to the sample, dry sample, like wheat, rice, and the other cereals like beans. So we add a 5 milliliter of water to the sample. Then close the door and shake it and vortex it. To show that the mixture is completely mixed with each other, and then put it for 5 minutes aside. Okay, in this step we add acetonitrile as an extraction solvent, and close the door and try to shake it and put it in the shaker. Okay, put it in the shaker. First try to shake the sample and then put it in the shaker. Set the time and the ramp. Okay, it's time, it's ramp, so you can start it. After 2 minutes, we will get out this falcon sample and go to another step. In this step, we add extraction kit into the sample in order to salting out the water that is in the sample. And it's good to mention that this extraction kit involves trisodium citrate, disodium citrate, and also magnesium sulfate anhydrous. Then after add it into the sample, try to shake it as quickly as possible. Unfortunately, I forgot to tell you that we use sodium chloride in this extraction kit. Okay, after adding extraction kit, we put the sample in the shaker instrument and shake it for about 2 minutes. In the next step, we put this sample in the centrifuge with 5000 RPM for 5 minutes in 5 degrees Celsius. Okay, after centrifuge the sample, we have this sample with a lot of solvent. 8 milliliters of the solvent will be removed and it will added to the cleanup kit, which includes primary secondary amine, C18 and magnesium sulfate anhydrous. After that we shake it and vortex it for about 30 seconds. Okay, after cleanup and vortex the sample, we push the sample again to the centrifuge for about 5 minutes with 5000 RPM and 5 degrees Celsius. Okay, after centrifuge the cleanup sample, in the final step, we should transfer 5 milliliters of the solvent into this kind of vial. Then we put this vial in the hot block with the temperature 30 degrees Celsius up to 35 degrees Celsius. Then after concentrate all of the solvent, we add 500 microliters of acetonitrile solvent, and then this sample is ready to inject to the GCMS instrument. I hope you will enjoy this video. Thank you for your concentration.